Hello friends, welcome to With My Plant. Today we will discuss about another hormone called abscisic acid. Abscisic acid and ethylene both are called as stress hormone. In particular with abscisic acid, um, let's start. So there are some important points regarding abscisic acid which we need to remember because these are very common questions. So the first important point is that abscisic acid, despite being commonly associated with inhibiting plant growth rather than stimulating it, it is a crucial hormone in plant physiology. It is widely present in various plant groups, including monocots, dicots, gymnosperms, and certain ferns. Then fruit and seeds. Typically, fruit and seeds typically contain the highest concentration of abscisic acid, highlighting its significance in regulating the process such as seed dormancy, fruit ripening. This is very important. Fruit ripening and seed dormancy is having two important or abscisic acid is having two important role in case of seed dormancy and fruit ripening. Then synthesized primarily in the leaves, abscisic acid is then translocated to the stem apex through the phloem, facilitating its systemic effects on plant growth and development. Now this is very important. When it is synthesized in leaf, it is moving through phloem and affecting plant growth and development. Okay, and sometimes it is synthesized in root, and there it used to move through xylem. So the name abscisic acid derived from initial discovery in high concentrations in newly abscised or freshly fallen leaves. That means recently fallen leaves, underscoring its role in leaf abscission and other physiological role. That means it has very important role in leaf abscission, and it was found through analysis of the freshly fallen leaves. Structurally, abscisic acid is a sesquiterpene. If we understand terpene, it can be hemiterpene, it can be a monoterpene, it can be sesquiterpene, diterpene. So hemiterpene is having 5 carbon compounds, monoterpene is having 10 carbon, sesquiterpene is 15 carbon compound, and diterpene is 20 carbon compound. So every sesquiterpene is having 15 carbon atom, and abscisic acid is among them. Notably, it possesses an asymmetric carbon atom, setting its part from the other, setting it apart from other plant hormone. So its molecular structure includes 6 carbon ring to which a single chain is attached, and the presence of the asymmetric carbon atom in carbon 1 results in two type of abscisic acid. Both are enantiomorphic. One is S abscisic acid and another is R abscisic acid. The naturally occurring abscisic acid is basically a ascorbic abscisic acid. Sorry, this is very important question. Okay, so these are some important points. Now coming to the discovery of the abscisic acid. So Torsten Hemberg in 1940 observed that dormant potato tubers and the ash buds contained inhibitor that counteracted the effect of IAA. That means IAA, some chemical was present in the dormant potato tubers and ash buds, which was antagonist to IA, a plant growth hormone. These inhibitors were found to be decrease the bud germination. That means they were causing bud dormancy. Okay. So, Igles and Waring, 1963, isolated an inhibitor from birch leaves that were kept under short day condition. Where this substance was applied again to birch seedling, it completely halted the apical growth. They termed this substance as dormin due to its ability to induce dormancy. Okay, so the first name for the chemical was dormin. Now, Occam and colleagues in 1965 extracted an inhibitor from cotton fruits and labeled it as abscisic 2. Another name was abscisic 2, which was isolated from cotton. Okay. Now, Conforth and his team in 1965 demonstrated that both Dermin and Abscisic 2 were chemically identical, and consequently they gave it an unified name called abscisic acid or ABA, owing to its well-known role in promoting abscission. Okay, so this is basically the discovery of abscisic acid. Now let's understand the physiological role of abscisic acid. The first and foremost role of ABA is the stomatal regulation. In further classes, we will understand the stomatal closing and opening, and the detailed role or the signaling mechanism of the ABA for stomatal regulation or stomatal closing and opening. So, ABA plays pivotal role in controlling the stomatal closure during water stress or drought condition. Okay, it achieves this by inhibiting the uptake of potassium ion by guard cell while promoting the leakage of malic acid. This reduction in osmotically active solutes causes guard cells to become flaccid, resulting in stomatal closure. So, a detailed mechanism will be discussed in further classes. Okay, and here it is to remember that abscisic acid is very important role in controlling stomatal regulation. That means the signaling of ABA will cause or will enhance the stomatal closure. Okay, next is your seed and bud dormancy. So ABA act as a growth inhibitor, inducing dormancy in birds and seeds across the various plant species. It inhibits lateral growth of bud, as observed in tomatoes. In temperate plants, ABA contributes to buds and seed dormancy by impeding growth. However, ABA level decline, growth resumes, often coinciding with increased gibberellin level. That means here ABA is antagonist to your GA. GA promotes bud and seed dormancy, whereas ABA inhibit. GA promote a bud and seed germination, whereas ABA promote bud and seed dormancy. Okay, next is your seed development and germination. ABA accumulates in developing seed embryos, either synthesizing de novo or translocated from leaf. It inhibits formation of germination enzyme in embryo and prevents vivipary. Exogenous ABA application inhibits germination in non-dormant seed, which can resume upon removal of ABA due to its inhibitory effect on germination enzyme and water uptake. So let's understand what is happening. One term is there, vivipary. Vivipary is a condition where seed germinates on the mother plant itself. or in other words we can say it will not have any dormancy period. There is no dormancy. It will just germinate in the mother plant itself. Okay, so because when ABA is not there, there will be such conditions, there will be non-dormant seeds which can cause vivipary, and if there will be ABA, this condition can be overcome. Okay, it will cause, and basically what is happening, ABA is having inhibitory effect on the germination enzymes. That's why this is happening. Next is your senescence and abscission. ABA is implicated as an endogenous factor in leaves and orange organ senescence and abscission. Exposed ABA application, exogenous ABA application induces leaf yellowing in various species. As photosynthetic activity declines, ABA production increases in senescing leaves once photosynthesis drops below the compensation point. What is happening? ABA is causing senescence and abscission. Abscission is the detachment of plant part from the mother plant naturally. Then senescence is the prior stage of plant part death or plant part death. Okay, this is the yellowing condition where and there will be dryness in the leaves. Okay, such condition can be induced through ABA, exogenous ABA application, or in natural condition also. If we see, in such stages ABA concentration is high. Okay, so it causes basically senescence and abscission. Next is your geotropism. Abscisic acid influences root geotropic responses with notable level detected in the maize root tip. Accumulation of ABA in the root tip influenced by light and gravity stimulates the positive geotropic response by acting as an inhibitor. That means basically what is happening, ABA influences root geotropic responses. That means root is elongated towards the gravity. Okay, or positively geotropic movement of root can be seen through because of the influence of ABA. Okay, so this is the physiological role. Some more are there. Next is important is your flowering. In long day plants, ABA inhibits flowering by counteracting the effect of gibberellin, while it induces flowering in short day plant. So basically ABA is antagonist to gibberellin. So gibberellin is enhancing flowering in case of long day plants, and ABA will inhibit the flowering of long day plants, but it will induce the flowering in case of your short day plants. Okay, next is your cambium activity. AVA halts the mitosis in vascular cambium as winter approaches. That means it will reduce the cambium activity, or it will stop the cambium activity by inhibiting the mitosis in the vascular cambium. Okay, that means it will indirectly it will reduce or inhibit the circulatory system of the plant by inhibiting water and food supply to the different plant part during winter. That means it will reduce the metabolism of the plant. Okay, next is your role in water stress. ABA plays crucial role in plant responses to water stress and drought. It enhances the plant tolerance to stress by and earning it title of stress hormone because of the stomatal closing and opening. It controls, as it is controlling the stomatal closing, that is why it can make the plant tolerant to your water deficit conditions. Okay, concentration of ABA increases in leaves under such stress, contributing to stomatal closure, regulating potassium and sodium uptake in gut cells. So we will understand this part in different class. Then counteraction of other hormones. ABA counteracts the effect of other hormones such as your IA, GA and cytokinin by inhibiting cell growth, amylase production and promotion of chlorosis. Okay. Its interference with calcium metabolism may underlie its antagonistic effects. And other effects, ABA inhibits cell division, cell elongation, induces parthenocarpic development in rows, increases cold resistance, inhibit gibberellin mediated amylase synthesis during grain germination and delays fruit ripening among the other effects. So if we see in as a whole, it is in reducing or it is inhibiting all the growth processes of the plant, and it is making the plant tolerant to adverse condition. That is why it is known as your stress hormone. Okay. So this is all about the discovery, some important point and physiological role of ABA. I hope you understood all the points, and some important points are there such as your stomatal closing and opening. We will discuss it in detail how it is acting on that activity. Okay. So this is all for today's class. Um, if you are not understanding some any point of this, you can comment below. I will try to make it simplified. Okay, thank you.